RBC count. Same chamber we'll be using, but here the counting chambers will be different. Okay. For RBC count, with smallest divisions, squares we measured. We calculated the number of RBCs present in the smallest squares, the smallest square. So here it is different. We'll see how to count that WBCs. So before that, we have to be acquainted with pipette. WBC diluting pipette, and fluid is also different. And we will find the composition of this fluid is different from RBC, and every component has different functions. We will ask in WBC. So try to memorize all these points. Okay, so what is there in WBC? How can you differentiate it from the RBC pipette? How can you differentiate WBC diluting fluid from RBC diluting? What are the purposes of this? While RBC counting, you are counting the intact RBC, but here it is different. What happens to the RBCs? So diluted blood. So here, where the RBC has gone, we will find there is no RBC. So where it has gone? So we will be asking these questions. That when you will be studying the composition and function of each component, then you will be knowing. Okay. So let's go to that room. Okay. So there is a normal range. If the count goes beyond this range, these are abnormal. So increased leukocyte count means increased production of leukocytes are termed as leukocytosis. Good. And what is the upper limit of normal leukocyte count? 11th. So if it goes beyond 11,000, we call it leukocytosis. Similarly, when the value goes down below the normal range, below 4,000 per cubic milliliter, then we'll term it leukocytopenia. So how leukocytes are counted, so we'll go through it, and before that we'll learn some theoretical aspects. I will highlight in between. Those are particularly important for your WBC. Okay, so where you have to take precautions, because this point is important. We will be assessing your skills in counting leukocytes. So there are certain points that we will be studying, we will be taking care of. So you have to be very careful at that points.
So today's class will be on total leukocyte count. So first, the size of the WBC is normally the number is normally in 1 micron liter, or we can say to 1 cubic mm. So the normal range of total leukocyte in adults, it is between 4,000 to 11,000. At birth it is 18,000, it can be plus minus 8,000. Till 1 year it is 6,000 to 15,000. For 2 to 6 years it is 5 to 15,000. And in 6 to 12 years of age it is 5,000 to 13,000. And in pregnancy it is up to 15,000. So you have to also know the normal range, okay, at the ages, okay. So after that, we will know when it is known as leukocytosis. Okay, so when the WBC count or the total leukocyte count it exceeds 11,000 per mm cube or meter cube, it is known as leukocytosis. So these are the conditions where you can find. So there are physiological conditions, there are pathological conditions as well. Okay, so what are the physiological conditions?
I ask you simply name one physiological condition in which leukocytosis is safe.
So
we may ask you two pathological conditions. So don't mix up. physiological and pathological.
So these are the mainly the physiological conditions. Like at birth, there is an increase in cell count. So normally, at that time also we see there is increase in leukocytes. So in pregnancy also it increases. Okay. Then in high temperature also increases. So there is going on a physiological basis. There is the mechanism. So there is one like a marginal presence of leukocytes around the margins of the vessels. So during when exercise or in high temperature, so that time the leukocytes shifts towards the central main circulation. So this is the physiological basis. Okay. So
you understood this?
This is
this point. Huh? Not follow.
So this is also known as shifting leukocytosis. Okay.
No, they couldn't follow. Repeat this.
So normally when when the vessel is like when we are resting,
In which condition?
sir, when we are in a resting phase, so normally the there is a stasis of like the flow will be less, so the blood cells will be towards the vessels, the endothelium. Okay, so when there will be like when we are there is increased movement or during exercise, okay, there will be a like dilation of the blood vessels. So at that time there will be rush of like the flow will be increasing. So that at that time the vessels due to dilation and the cells comes towards the main circulation, the main flow. Okay, so this is not at that time also there will be increase in WBC count. Okay.
We may ask you the why during high temperature TLC is raised. What are the physiological basis? We may ask you why during pregnancy is leukocytosis. What are the physiological explanations? So you have to answer that. So I am trying to inspire you to ask questions. You have to know, no? So without asking question, how it will clear limited time. So within this limited time, we have to cover so many things. So if you have specific questions, sir, I have to know this, then it can be answered. So you have to be incredibly mind. Know that is this. Know how your competency is written as A and AH. Right? Knowledge at the center, how. Knowledge is this, in pregnancy, it is great. But why it is great? This is the how. So knowledge at the same time, how. That so that you can explain the phenomenon.
So in physiologically we find in these conditions, and there are also pathological conditions like in infections, in malignancies like leukemia, so where there is increased production of WBCs, and in severe hemorrhage. Like in infections, what happens mainly? Like what happens that time? There is an immune response. For that, there will be increased production of WBCs, or there will be the bone marrows will be mostly activated, and the mature lymphocytes will be acting on the infections and phagocyte it, and then this is our defense mechanism. By this way, the WBCs counter the infections. Okay, so that time also in there will be in pathological. This is pathological condition where there will be leukocytosis. Okay. And in malignancies there is increased count of the WBCs. Okay. Then in severe hemorrhage also we see. Then we have to know when it is called as leukocytopenia. So when the total count of the WBCs or the leukocytes is less than 4,000 cells per cubic mm. So these conditions like in typhoid mainly, and viral infections like hepatitis, influenza, then in protozoal infections like malarias. So here what happens mainly, there is an immune response. Okay, so during the immune response there will be release of the histiocytes. And then these histiocytes, what they do is phagocytes our normal cells, that is the WBCs, RBCs, platelets. Okay. Also they suppress the bone marrow. So that time there is neither there is production of the WBCs, nor there will be our defense mechanism to act on the infection. Okay. So this way mainly there will be decrease in WBC count. Okay. So this is the main mechanism behind the leukocytopenia in infection of the bacterial, viral and protozoal infections. Okay. So in megaloblastic anemia also we will see normal decrease in leukocytes, and in drugs also, like in aplastic anemias and in radiation. So in these cases also there will be decrease in total leukocyte count. Okay. So the methods today, no, not written. So there are also like drugs that can also cause bone marrow suppression. Okay. So we have to know the drugs also, like methotrexate. Okay. Such kind of drugs, the cancer drugs. Okay. Those drugs also suppress the bone marrow, and this will cause formation of decreased WBCs. Okay, written. So there are two methods, the visual method and one is the electronic method. So today we'll be seeing the visual hemocytometer method. Okay. So here first we need a blood component and cell, which means which is describing the term the hemocytometer. Okay. The meter is used for the measurement. Okay. So it is an instrument used to count blood cells. Okay. So here we need in samples the EDTA, then our capillary blood. So then the one microscope, one hemocytometer, now one WBC diluting fluid. Okay. So these things, the hemocytometer you already know it. Okay. The Neubauer slide, and then the cover slip, the RBC pipette, the WBC, you already know it. Okay. In the previous classes we have discussed this. Okay. So you know the Neubauer slide. This is a thick glass, the H-shaped. Okay. There are two slides which are the central horizontal bars. You know it, na? Okay. And the chambers where it is located.
They are going to do this procedure. If you require this explanation of this, no. Okay. And now this is important.
Actually, this is the slide. Okay. So this we have on the last class of the RBCs we are focusing on the central square. Okay. The central square is the which square? The central square which we are focusing, that was the for RBC counting.
Which are the boxes?
Which 5 boxes we are actually count for RBC?
It's like center, then corner. Right?
Is she correct?
Yes, sir.
This is the area for counting RBC. And not all the, only 5. 1, 2, 3, 4 and 5. Now go.
So this is one large box, na? This is one large box, and this is one small box.
What is the distance from here to here? From here to here? Here to here?
So this is the one large box, and this is one small box, and for RBCs, this is the one smallest box. Na? You have to know this. Okay, the difference between large, small, and smallest. So similarly, this is the length and the breadth is 1 mm into 1 mm. So you have to calculate the area. If you calculate the area of this small box, This will be
What is the area of this small box? Area and volume of this box, single box. Area and volume first. What is the side? 1/4. From here to here, 1 mm. Now you are dividing 1 mm into 4 divisions.
4 divisions.
So this area is 1/4. So 1/4 into 1/4 into 1/10. This is the volume. Suppose this volume contains 5 RBC, 5 WBC. How many? What would be the value in per cubic millimeter? Convert this value into per cubic millimeter. Do it. Number of WBC is 5 in one small division. This area. 1/4 into 1/4 into 1/10. This is the volume.
Volume.
And in this volume, we have only 5 RBC. WBC, what would be?
Count per cubic
Cubic millimeter. Simple. Unitary method, you do it. In 2 small squares, there are 12 WBC. Again, it is simple for you. In 12, in 2, 1, 12. In 1, it will be 6. Averaging. Then you count it like that. It is as simple as that. And then after that, you just multiply it with the dilution factor. We are coming to that later.
So in a WBC square, you have now seen. I think you will be remembering which is the WBC square.
Dipankar, one more thing. During practical exam, doing practical, what you will be doing, calculating number of WBC here or here?
only in one book.
In all the boxes? In one box? No, in all the boxes. Okay, sir didn't tell us till now. বলেছে স্যার? বলেনি। তাহলে কি হবে স্যার? সবগুলো বলতে হবে?
Yes, sir.
সবগুলি করতে হবে। তাহলে সবগুলো যদি আমাকে করতে হয়, তাহলে এই যে জায়গাটা, এটার এরিয়া কত, ভলিউম কত? Now calculate the area of this and the volume of this. Area is simple. 1 into 1. And volume is into 0.1. So this is the volume. So this volume, this much volume contains this number of WBC. So what would be the value for in per cubic millimeter? Again, it is simple. So you have to know how many squares you have counted, what is the volume of that area, and what is the number of WBCs there. So if you know this, why I am telling this? Because during WBC or during problem solving, this can also be a problem solving question. In 5 such squares, 1, 2, 3, 4, 5, or 1, 2, 3, 4, 5, any 5. Number of WBCs are this much. Can you count the total count per cubic millimeter? Okay, I am giving the figure. There are 25 WBCs in 5 small squares. Area, height is known, common for all. If you know the area, in this area how many are this, so what will do the volume, in this volume how many, same, so then convert it into per cubic millimeter. So, everybody will be asked. I will be asking you in WBC, I am telling you. So be prepared for that. I will be asking, I may tell you in 5 divisions, 5 such squares, there are total number of this. I may some others, there are 3 small divisions, there are a number of WBCs. This much, what will be the value? Dilution factor is this. I think everybody will be able to answer. Good. Next. Manusha, ঠিক আছে তো?
So, in total, there is 4 into 4, 16 small squares,
শিখবো তো?
okay, in a WBC square, okay. So, the 4 corners of the WBC square, okay, we count the 4 corner squares for WBCs, okay. Then, this is a
তোমাদের কিন্তু কাউন্ট করতে হবে 4, আবার একটু ব্যাকে যাও, এটাও কাউন্ট করতে হবে, এটাও করতে হবে, all these 4. Can you tell us what would be the volume of these 4 corners? 1 into 1 into 0.1 into 4. Simple. 1 into 1 into 0.1. This is 1 corner. So how many corners you have? 4. So multiply it by 4. So this is the total area. So in this total area, number of WBC is this much. So what would be the value in per cubic millimeter? And this you will be getting in diluted blood. If dilution factor is, we will be telling you what is the dilution factor. So you will be multiplying that value with the dilution factor. Then you will be getting the original blood per cubic millimeter of original. Next.
So, we have known the RBC pipette.
Pipette.
So,
WBC diluting pipette.
so this is the WBC pipette, okay. So here, how will you identify this is a WBC and
আর একটা বার
not
করো,
RBC?
তিনটে বার করো।
White bead.
Yes, white bead, na?
আর
The
একটা বার
presence of white bead and
See,
markings. Yes.
look at the marking.
What is the marking? What is the first marking?
What is the first marking? 0.5. Then
Then
1. And at the above, at the top of the
11.
1.1.
11.
Okay? So now see, there are how many? This is one identifying mark.
So the size
There,
of the bulb
there is the bead, and
size
instead
of the
in
bulb is-
RBC it is 101, but here it
10
is.
units. Na?
This is the
So in
difference.
RBC it was?
Will you go through this?
Yes. It was 100.
Is it
Huh?
required?
In RBC it was 100. So here it is 10.
One condition was it will not move. Moving, no? One condition was it will not move. If the pipette is not dry, if it's wet, it will stick. Okay? It is not problem. Okay? You have seen it in RBC class also. WBC pipette. Difference between pipette and tube. Anyone knows the difference between tube and pipette? What is RBC tube? Pipette and tube. Difference there. Why it's called RBC pipette? Sorry, WBC pipette, not WBC tube. Anyone? Why it's called pipette? Tube. Because tube has only one end open and one end is closed. Here both end is open. This is also open. This is also open. Okay, that's why it's called a pipette.
So it has graduations till mark 11. Okay. So now the diluting fluid. So
See the composition. Learn by heart.
so in composition there is glacial acetic acid which is 2 ml. So what does it does?
What is the function of this glacial acetic acid? Sir will tell once.
So
Don't repeat. You will be telling one take.
so it helps in to hemolyze the cells. Okay, it helps in hemolysis of the other cells.
Hemolysis means which cells? RBCs. There will not be breakdown of WBC. Why? If acetic acid can break down RBC, why not WBC? It can. The WBCs has no preventing mechanism by which it can prevent acetic acid from lysis of the membrane. So there will lysis, and the nucleus will become exposed. The nucleus will become, it will not go away. Okay? Next. Now see, what is the use of gentian violet?
So what is the composition of gentian violet? It is 1%.
1%, how can you, if I ask you, prepare a 1% solution of gentian violet. So gentian powder is given to you, and distilled water is given to you. Distilled water?
Yes, sir.
How you will prepare it?
100
In 100 ml, how much? 1 gram. See the confusion. Somebody is telling 0.1, somebody is telling 1 gram. 1 gram. 1 gram. Are you sure? Not sure. 1. Now you are sure it is 1. Okay. So this way, weight by what is this? What does it mean? W by V. Suppose the gentian violet, it is not in solid form, it is in liquid form. Then what would be written in between, within the bracket? Volume by volume. In that case, how much you will be taking? 1 ml. Good. So there is, it is prepared as follows. What they have taken? Glacial acetic acid 2 ml. Then 1% gentian violet 1 ml. Then distilled water 97 ml. So 97 ml, 1 ml, 98 ml, and 2 ml. Ultimately, volume is 100 ml.
So the use of gentian violet is?
No.
Sir?
Go to next slide. Now, you tell me composition of WBC diluting fluid.
glacial acetic acid.
How much?
2 ml.
Okay.
Distilled water and 1% gentian violet.
How much?
1% weight by water.
How much?
Oh, that is 1 gram in 100 ml.
How many? After ultimately, I have to make 100 ml, no?
Yes, yes.
So 97 ml I will be taking water, and that glacial acetic acid you have taken.
I say 1%.
Glacial acetic, how much you have taken?
1 ml.
How much? 2 ml? Huh? Or 1 ml? 2 ml. So remaining 1 ml. That would be obviously it would be gentian violet. দেখেছো বাবা আগেই বলেছি এটা লার্ন ইট বাই হার্ট আবার নিয়ে যাও ব্যাক এগেইন আই উইল আস্ক 5 টাইমস করে পড়ো ছোটবেলায় যেন আমাদের পুরো শেখানো হতো স্পেলিং APPLE অ্যাপল APPLE মুসো তোদের এই হাল হয়েছে What is the function of acetic acid, glacial acetic acid? Why it is used? Huh? Hemolysis. What does it mean? Breakdown of blood cells. Okay. So you learned the composition. Okay. Next.
So the use of gentian violet is we use it for staining the nucleus okay, the nucleus of the leukocytes. So it is diluted in the ratio of 1:20, okay.
শুধু নিউক্লিয়াসটা বেরিয়ে আসবে ওটাকে আমি
And
they will
with the
not
use of
see the
diluting
whole
fluid,
WBC.
the cells are counted under the low power, that is 10x, okay,
হ্যাঁ
by using the
nucleus
microscope
stain
on the
করবে।
under the counting chamber. And the number of cells
যে ও শুধু RBC
in
করবে এটা
undiluted
করবে না।
blood are reported per
কনসেন্ট্রেশন আমরা
cubic mm
টা দিচ্ছি
of the whole blood.
আরবিসি টা আরবিসি
Okay.
আর একটু বেশি কনসেন্ট্রেশন হলে তাহলে আমার WBC লাইসেন্স
তোমার কনসেন্ট্রেশন কত দিচ্ছ তুমি
2%।
2%।
2%। একটু বেশি কনসেন্ট্রেশন
দিল কিন্তু আমার মেইন এটা হলো কি আরবিসি আমি অ্যাকচুয়ালি কাউন্টিং দ্য ডাব্লিউবিসি আমি কাউন্টটা করছি কি আমার ফিল্ডে আসছে শুধু নিউক্লিয়াস গুলো ওরা কিন্তু সেলের সাইজ এটা পেরিফেরি দেখতে পাবে
ছোট ছোট ডটস দেখব
ডট দেখবে মানে নিউক্লিয়াস গুলো দেখবে নিউক্লিয়াই গুলো দেখবে
হ্যাঁ
Okay. Huh?
যে মাইক্রোস্কোপ এটা কেন আলাদা করে লিখলো ওয়ার্ডটা মাইক্রোস্কোপ চার নম্বর পয়েন্টে
হয়েছে
अच्छा नहीं हुआ नेक्स्ट स्लाइड प्रोसीड्यूर
Procedure, you know.
just similar to RBC count यहाँ पे till 11 only instead of 101 okay
मैं ये परेक्टा next no before that back again forward how much blood is diluted how many times how many times blood is diluted 20 times confusion
200 times.
আরবিসি 0.5 নিয়ে 101 করেছিলাম কত টাইমস হয়েছিল এখন আমি 0.5 নিয়ে 11 করছি তাহলে কত হবে 20 টাইমস তাহলে আমি কত দিয়ে মাল্টিপ্লাই করব ডাব্লিউবিসি কাউন্ট করার সময় ওই যে ডাইলুটেড ব্লাডে এতটা তাহলে হোল ব্লাডে কতটা ইনটু 20 ওই যে হিসাব করে বার করেছিলাম এটার সঙ্গে 20 দিয়ে মাল্টিপ্লাই করব দেন ইউ উইল গেট দ্য কাউন্ট in per cubic millimeter of blood.
So I have already said there are markings, okay, and the point of identification. How will you know this is a WBC pipette?
So here there are 2 points. You have to take precaution. We will see it during your exam. Whether you have taken blood from here to here. This is one point. If it is less than this, if it is more than this, your marks will be deducted. If we find at the tip of the pipette, in the outer surface, there is some blood, then mark will be deducted. It has to be wiped. Okay, then so you have taken the blood correctly. So it will depend, some marks will be there. Okay, after that what will be there, what we will be doing? We will drawing diluting fluid, WBC diluting fluid. Okay, up to this mark.
So here, how will you like reduce the excess blood? No. So here you cannot use an absorbent material. Okay, so what will happen if you use an absorbent material? It will suck the fluid portion and there will be more concentration. Okay, so you cannot use here an absorbent material like cotton or a gauze. Okay, you have to tap it. Okay.
But here you have carefully wipe excess blood outside the pipette by using cotton.
Sir, here it is written, sir.
Huh?
But actually we cannot use, sir. Okay.
So you got the process, got the point.
So it will make the concentration high inside the pipette. Okay. So the fluid will be sucked. Okay. So then we will take the diluting fluid till mark 11. Okay. Then we will again mix. We'll keep it in horizontal position for few minutes. We'll mix. Then yes. So then we need to discard. How many drops? 2 to 3 drops. Yes. So after that we will go for charging. Na? So where will you put the drop? So before that, you need to ensure the Neubauer chamber, the cover slips are dry and clean and not. Okay. Before that, you need to ensure that, and after that, you need to focus the chamber under the microscope. Okay, in 10x. Okay, that time, after that only, you will be placing the or charging the chamber. How will you charge? Edge of the cover slip. Yes. So it must be at an angle of at the edge. That time what will happen?
First 2 drops are discarded. Because only mixing occurs in the bulb area. Okay. The fluid and the blood is not mixed in this part and pipette ka end part portion mein this is not mixed. That's why we discard the first 2 drops.
So here you need to have a precaution. The chamber must not be an undercharged or overcharged. So how will you know it is undercharged? So if the drop is not sufficient or it is not in position, so it will be not evenly distributed on the chamber. Okay, like half of the chamber is filled or charged, the rest is not. Then it will be undercharged. How will you say it is overcharged? Yes, when it will be overflowing. Okay, that time. So if you don't you do it in the rules or the steps which we have said to you, so there might be errors also, like there will be presence of air bubbles. Okay, so with proper precaution you need to take the blood sample and the diluting fluid. Okay.
Sources of error. One source is pipette error.
Yes.
Another source is dilution error. Another source is chamber.
Yes.
That's not good.
Chamber error. Another source is calculation, statistical calculation error. And another is field error. Okay, so you have to take care about these errors. Chamber, you forget about chamber error. Okay, so here it is not, their possible error is field error. Field error, statistical calculation error, and dilution error, pipette error. So these are the points we will be looking into. Suppose you are having 20 marks. We have 40 marks for you in your coming exam. You have to perform 2 experiments. And you may either, some of you may get WBC count, some of you may get RBC count. Okay, so it may every experiment will be having either 20 marks or 25 marks, depending on since we'll divide or already divided 20 into 20. So then it is 20 marks. Okay, so these 20 marks we will be dividing into this. How we will give 20 marks? So we will assess you at these 4 points. So you have to be very careful about this.
So in focusing, first you have to focus the slide on under 4x. Then for WBCs we have to go to 10x. Okay. So
for 5x it is for the survey purpose. What is the condition, overall condition?
yes.
Then you shift. After focusing through the survey objective, that is 4x, 5x,
4x.
4x, then you shift the 10x. Then you focus it and don't break the hemocytometer. Because you will be lifting the stage. আমাদের lifting না না ওই যে আমাদের মাইক্রোস্কোপগুলোর আমরা যদি ঘোরা এটা কি আপনার বডি পার্টটা নিচে নামে
Stage, sir.
স্টেজটা আপ ডাউন করে সো
Adjustable.
you may break. If it is lifted in excess, then it may break. The hemocytometer may become broken.
So in counting, we will mainly use the similar method, okay, in which we used in the RBCs. That is the left and the lower, okay. So follow one rule, okay, whichever you are preferring, okay. If you are counting the left and the lower, always stick to it, okay. This is to avoid any further recounting of the cells, okay. So similarly, we will go in a zigzag fashion, okay, like from left to right, and then right to left, and then left to right, then it will be continue, okay. So this is a large box now. So we are if we count the left lower box, then we will be counting only the left and the lower portion, okay. So you can count here. So can you count here how many cells are there?
What is the WBC count of the patient now? Count it.
Why we are using 10x for counting WBC, but why we are using here 10x?
Why not 40x? To count number of RBCs here in the smallest division. If you don't focus it under 40x, you won't be able to count it. Demarcation will be not clear to you. So that's why we have used 40x. And this total area is under focus. You will find, this total area you will find there in the field. Under 40x, you will find the total area. You have seen it. Okay. So if you use for this 40x, you will find only this portion under the focus. When you are studying WBC, under 40x, you can see only this area. So under 40x area, you will be able to see only this area. This will not be, you won't be out of focus. So if you bring it under 10x, then all these boxes will be available to you. And since their number is less than RBCs, so it will be very easier for you to calculate, to count. Very fast you can calculate how many. So now you see, I am giving you 30-second time, 20-second time, count. Same principle. Touching upper border, touching right border, excluded. But touching left line and lower line included. Now you count how many and write it down. Individual, don't copy from others. You know this area. This area is known to you, isn't it?
Large square or small square? This is small square.
Large square. This is 1, 1, 1, 1. All sides are 1.
That side is already told.
1 into 0.1. This volume contains this mass WBC. Dilution factor is 20. Now you tell us what is the value. But then they will be knowing the calculations.
So how many cells are
You
here?
count. How many cells you counted?
What is the square also?
You tell me the figure.
19, 17, 18.
Cross is given. Touching upper line and right border is excluded. And all others are included. So now count how many are there? Who said? More than 19 or less than 19? Who? You come here. Are come. You have to learn, no? No, come. Count. Go and there. Go there. 1, 2, this way. Show kore.
Show. That cell you will count or not? That cell? This one, yes. This one. Yes.
Have to count. 1, 2, 3. Ha.
5,
Ha.
6,
7, 8, 9,
What about this?
10, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19.
Now you are convinced.
Sir, मैंने count नहीं किया था.
So there are how many cells?
19.
Yes.
Okay.
Any no confusion, na? Any
What is the
confusion here?
This one.
19
Whether
cells.
this is touching this, then it is the property of this. So it will become over.
It's touching this side.
No, it is touching here. When I will be coming here, so this is touching my line, so it will be hit, counted here.
We won't count from this side.
When you will be coming here, it is already being counted, so I will not consider this. One cell cannot be counted twice. Got it? Any doubt? Any doubt about the count number 19? It is 19 or more than 19 or less than 19? 19. So, okay, you are confident. Okay, now you calculate. TLC बताओ. First, ये area लिखो. कितना है area? 1 into 0.1.
Large box you have counted, na? But the calculation is about the 4 boxes. The calculation is about 4 boxes. So you have calculated in 4 boxes. So you should count all the 4 boxes. So this is 1 box. So more 3
This
boxes.
is one box. So if you count 4 boxes, the number will be increased and area will also be increased. Then this mass area contains this mass. What would be in per cubic millimeter? Same.
This is the calculation, okay?
So this is the calculation. So what will be the n? What will be the n? Number of cells. Number of cells in?
Why this 4?
Number of cells in where? Yes. So that was the one large box. So you have to count 4 boxes. So that time the total n will be you will be getting. So then after that only you will be calculating. Okay. Did you understood? In RBCs also we have counted only 1 box. So more, how many boxes left? Yes, RBC, more 4 boxes was left. So we have not counted that. So after counting that only, the total n will be, you will be getting. So after that you will be getting the calculation. So here also similarly, we have counted the 1 large box, more 3 boxes are left. So after counting that only, you will be getting the total number of WBCs per cubic mm.
Dipankar,
Sir.
during exam, we may not ask you to calculate all the 4 boxes. We may ask you to calculate, to count the only 1 large box. 1 box instead of 4. Then you calculate. Because you are 50 students, more time will be required. So from 1 box you will be calculating. So in that case, what will be your formula? Suppose, what would be the formula in that case? Write in your copy the formula.
So we will just remove the 4.
Then what is the answer?
And by n into 1.
We will count only 1. Put down multiply by. একটা ভ্যালু তার দ্বিগুণ করবে all the boxes might not contain the same number of WBCs, right? So same shortcut তাহলে আমরা 4 দ্বিগুণ করে দেব
ফান্ডা হয়ে গেছে ফান্ডাটা তো করে নিয়েছে বুঝে গেছে ব্যাপারটা where you understood the matter only thing when you will be asking immediately you have to calculate it problem solving so this will be the problem solving questions for 2 marks 3 marks so be prepared for that see these are the tricky questions you understood are you everybody prepared for this she is not prepared I'm sure see she is smiling she is not prepared make it clear from sir now only formulator likhe dekha in that case instead of 4 you have counted only one corner there are 4 corner 4 large boxes no so you have counted only one box and from that you have to count what in that case what would be the formula
So there is another method. It is known as electronic method. Okay. So we don't usually do with the electronic method. Okay. So there is another method. That is electronic method. So we will not be doing this method here. But for your information, you can know it. Like it is an automated method and it has some advantages like it is easy and rapid method, it is time saving, and there are a very large number of cells which can be counted rapidly, there will be high precision level here, okay. So as well as it has some disadvantages also, so like it is costly, there will be calibration error, the nucleated RBCs are counted as leukocytes, the platelet counts can be counted as leukocytes, okay. So mainly
calibration error. Calibration error. What is this?
Markings are a bit distorted.
The value given by this method, whether it is correct or not, how should I know?
so it's set a standard, right?
There should be a standard method. So by the standard method, I will be counting. And this is the standard method you are using. Now you will be doing, so by standard method I counted 5,000 per cubic millimeter. And by this automated method, it has also gave some reading and it result was instead of 5,000 it is 8,000, not acceptable. So this is not standardized. The value should be within the machine will tell, it will give 5%, 2%, 1%, this much of error. Error, variation from the standard method. Another, you have seen the blood pressure monitor, electronic one. Have you seen it? Huh? So did you measure twice or thrice, same person, you are measuring, same person, same position. You have taken one measure, again you have repeated. Are both the results same? So it may vary. It is a variation, not it is because of your fault. This fault is with the machine. It is some variability. Another fault is individual variation. Say by sphygmomanometer. Same person lying, I have asked you, measure his blood pressure. So you measured it. Again I asked you, again you measure it, you will not find identical value. These are individual variation. Variation of the investigator. In titration, while you have done titration in chemistry laboratory, how many readings you have taken? 3 reading, then you take the average. So here also you have to take 2 or 3 readings, then you have to make the average. because all these variations are due to your observer variation. And person to person again will vary. You have measured this much, when she will be coming, she will measure a different, she will give a different value. These are the person to person variation. So who is correct? Your measurement is correct, or her measurement is, you are measuring blood pressure of sir. So you have measured, she has also measured, which one is correct? How should it be assessed? Average. She doesn't know actually. She just knows my normal pressure is this. So she gave some 120 by 80 millimeter of mercury. What is the normal value of blood pressure, mean average blood pressure?
20 by 80.
20 by 80. Hello, normal blood pressure?
So 120 by 80. 120.
120 by 80. 80 by 120. 120 by 80. 2020 match चल रहा है भाई. I told you repeatedly in practical class, in theory class, whenever you tell the value, please mention the unit. See the concept. That's why I asked.
What are the other uses
Bad.
of Neubauer chamber? We have done the RBC count. Now we are doing the WBC count. So other than that, platelets can also be counted. The cerebrospinal fluid cells that can also be counted, and the sperm count. Okay.
আমরা যখন রিপ্রোডাক্টিভ সিস্টেমে যাব তখন ওই স্পাম কাউন্ট গিয়ে নর্মাল ভ্যালুস ট্যালুসগুলো কি সেগুলো আমরা আলোচনা করব, কখন কমে যায় কখন বেড়ে যায়.
So this is the machine.
This is the
automated machine where the cells are counted.
Not only RBC count, WBC count, all the counts will come. Okay.